Hello dear everyone, how do you do? This is Mr. Karthikeyan, mentor in NEET, Bowskin and Zoology subjects. Welcome to my video session. In this video session, I want to explain to you the structure of Dicot stem. So on the board, schematic and diagrammatic presentation we have. This is the entire structure of Dicot stem, TS, transverse section, or the anatomical structure of Dicot stem. Let's discuss how the tissue arrangement is there in the Dicot stem internally. In last video I have been explained Dicot root, so you could go through that and learn well. In this video, everybody listen carefully and learn well. This is also essay answer, carries 8 marks for intermediate public examination, Andhra Pradesh, and also PUC, Karnataka, NCRT in our India, different states wide, and also MCET and NEET bits also will be raised. So keenly you could listen and observe and try to find out MCET bits and NEET bits too. Okay, done. Then same like root structure. In this Dicot structure arrangement, main parts are there. Number 1, epidermis. Number 2, cortex. Number 3, stele. Epidermis, cortex, stele. As usual, like dicot and monocot root. And let's see the difference between root and stem. Right? Okay. So first one, epidermis. You knew already. Epi means outermost layer or upon. Dermis means layer. So epidermis is outermost layer in the stem, TS. On the epidermis, thick cuticle is present. And also stomata here and there can be seen. The main function of a cuticle, waxy coat or layer is prevention of loss of water, nothing but transpiration. Then epidermis is a single layer. Cells are rectangular, closely or tightly or compactly packed, no intercellular space, single layer. Here and there, epidermal outgrowths are seen. These epidermal outgrowths are called trichomes. But in root, that is known as root hair, unicellular. Here these are multicellular trichomes. The main function is directly, indirectly protection and also prevents the loss of water from the stem. Then the epidermis main function is giving protection to the innermost tissue of the stem. Right, then we shall go for next one. That's the information of epidermis. Okay, then coming to cortex, the tissue which is distributed or present between stele and epidermis is known as the cortex. As usual, cortex is made up of 3 main subregions. Number 1, hypodermis or exodermis or outer cortex, then general cortex, then endodermis. Hypodermis or outer cortex or exodermis, general cortex, endodermis. Same as usual, root. In stem also cortex has 3 subregions or subparts. First one, hypodermis, just below the epidermis, 2 to 3 or 3 to 4 layers of cells are arranged. These cells are collenchymatous. Nothing but the cells are collenchyma, not parenchyma, collenchyma. So cells are thick, intercellular space is absent, So the main function of this collenchymatous hypodermis is giving mechanical strength and flexibility, rigidity to the stem. So it prevents maximum without breaking the branch or stem against the external factors. So that's called mechanical support or flexibility and rigidity. Done. Then followed by parenchymatous cells arrangement. Many layers are arranged. Those cells are parenchymatous cells. Huge intercellular space is present. Thin cell wall we could observe in the cells is known as general cortex or simply cortex. So this general cortex main function you knew well, storage. So same like root. In roots, general cortex is enclosed the number of glands, but in the stem, dicot stem, angiosperms dicot stem, resin canal is enclosed by parenchymatous cells. Resin canal or resin duct. So this resin canal or resin ducts enclosed by glandular cells. This is resin duct or resin canal. These are glandular cells. So it secretes resin, tannins and etc. Those are secondary metabolites. In 10th standard you might have been learned. Tannins, latex, what it is called, alkaloids, etc. Gum. These all are secondary metabolites of the plants. Presence of this resin canal or resin duct is the characteristic feature of or main character of angiosperm dicot stem. Very important for competitive purpose. Remember everybody, dear students. Then followed by endodermis. In root also we can be seen endodermis. Here also the endodermis is a single layer. Casparian strips or lens thickening can be observed. Casparian strips commonly it is called. And cells are barrel shaped, not rectangular, barrel shaped cells like drum like cells. Casparian strips are present. Main function is giving protection to the remaining tissues present in the stem. And the main characteristic feature, competitive point of view, endodermis cells, they possess or contains starch grains. Generally starch grains are present in chloroplast, right? But in endodermis starch grains are present because endodermis is also known as starch sheath. I repeat once, endodermis is also known as starch sheath in dicot stem. Remember well. Then this is about the information of cortex, hypodermis, general cortex, endodermis and their characteristic features. Over. Right? Done. Next, coming to stele. What is meant by stele? There everybody, same like in root, pericycle, vascular bundles and medulla. These three combination together called stele. The central conducting part of the stem, same like root. But here the arrangement of the tissues are different. Let's see. Yeah, done. Generally, pericycle is single layer in root, but here pericycle is multi-layered. In root, pericycle is made up of parenchyma, but here pericycle is made up of sclerenchyma. There is no continuous ring, patches wise pericycle is formed. That is one of the characteristic feature. Small patch, then the gap. I will tell you soon what these cells arrangement and structure is called. Then pericycle or sclerenchyma, then gap, again, etc. So alternatively, pericycle tissue is arranged. That is one of the characteristic feature. Done. Then exactly followed by pericycle, these cells arrangement is called phloem. Main function is transportation of food. Then followed by phloem. These stripes are known as cambium. This is one of the characteristic feature in the stem. Then followed by xylem. So, phloem, next cambium, xylem. Phloem, next cambium, xylem. This is known as vascular bundle. Arrangement of the xylem and phloem together called vascular bundle. Between xylem and phloem, cambium is present because this vascular bundle is known as collateral conjoint open type. Remember everybody, this is one of the competitive bit. Between xylem and phloem, cambium strip is present because it is called collateral conjoint open type of vascular bundles. If cambium will be absent between xylem and phloem, then that is called closed type. Collateral conjoint closed type. Let's see in other topics where we can find such type of closed type. But this is, remember everybody, conjoint collateral closed type. And here we could observe the xylem arch arrangement. This is called metaxylem, larger one. This is called protoxylem, smaller one. I already told in a previous video while discussing the Dicot root. So metaxylem present towards peripheral, protoxylem present towards inner side, nothing but medullar pith. Protoxylem, metaxylem, protoxylem, metaxylem. Such type of arrangement is known as endark. So this arrangement is called endark. If protoxylem present towards periphery, that is called exact. Competitive purpose, remember very well, dear students. So here, collateral conjoint open type of vascular bundle and endark condition. Done. Then coming to next part, medulla. The huge well-developed parenchymatous tissue arrangement is known as medulla or pith. It is well-developed. The main function is storage of the food material. And then sir, what is the structure? From the medulla, special cells are extended. What is this structure? Extended between vascular bundles, nothing but, dear students, from the pith or medulla, the extension of the cells, this structure is known as medullary rays. So this medullary rays helps in the lateral conduction in the stem. Xylem and phloem, they transport or translocate the food materials bottom to top and top to bottom, unidirectional or multidirectional. But this is dicot stem, right? Secondary growth will be seen, because for that secondary growth, the lateral conduction of the food must be, the lateral conduction of the food controlled by, taken by, fulfilled by, done by medullary rays, parenchyma tissue. Extension of parenchyma tissue from medullar pith, dear students. Then this is cambium, I have told you. So the cambium present between xylem and phloem is known as intrafascicular cambium. Remember, intrafascicular cambium. During the secondary growth, that is one more topic, lengthy topic, we will discuss in one more video, but the basic I am telling you, everybody listen carefully. This is intrafascicular cambium. From the medullary ray, few layers of parenchymatous cells, they are differentiated into meristem cells. Those meristem cells again differentiated into cambium cells. So then, intrafascicular cambium and interfascicular cambium, these both type of cambium together form cambial ring, dear students. That cambial ring is the responsible for secondary growth of dicot stem, which increase the girth or width of the stem, nothing but enlargement of the stem. That is called secondary growth. So dear everybody, this is the information regarding dicot stem, trichome, cuticle, epidermis, hypodermis, general cortex, resin canal or duct, starch sheath or endodermis, starch grains, multi-layered pericycle, medullary ray, huge and well-developed medulla or pith, vascular bundle, pericycle followed by xylem, followed by cambium, followed by xylem. Then this arrangement is known as conjoint collateral open type. This is metaxylem, this is protoxylem. So I hope everybody understood this topic well. I shall meet with you in one more topic in one more new video session. Till then, thank you. Signing off. This is Mr. Karthikeyan, mentor in a NEET, bone skin and zoology subjects.